So if you want to look at it, it's the power of life or death in your protein. So please welcome Tca Hecht.
Can everybody hear me? Are we okay? Yeah, okay. So it's really hard to get up and give a talk when you've been introduced as somebody who's going to talk about the power of life and death, but I'll do the best I can. So, okay, so this is the title I originally submitted when I was asked to speak here, but I'm going to simplify things a bit, okay? I'm also going to use this as an opportunity to do something a little bit different from what most iGEM presentations are about. I think most of you, when you're presenting your research on posters, are going to present the details of the experiment, but I figure as, you know, one of the invited speakers here, I'm allowed to be a bit more philosophical and take a broader picture. So you'll get lots of data at the end of the talk, but let me start with a much broader introduction. So let's think about life, okay? And again, is it echoing? Is it okay? Is the sound okay? Somebody tell me the sound is okay? Okay. So we're going to think about life. So these are ants. You didn't expect me to start with ants. You expect me to start with E. coli and yeast. But these are ants. And this is a colony of ants that was taken somewhere in Africa. And ants, in a colony of ants, they all have common ancestry, right? So all of the ants have the same genetics. Basically, there's a queen ant that is the parent to all the ants. And because they have common ancestry, they have common genotypes and common phenotypes, and they're all basically the same, right? And if you want to look for something with alternative properties, different genotypes, different phenotypes, you want to look for alternative ancestry. Well, how do you do that? You go to another continent, okay? So here's a colony of ants in another continent. One picture is from Africa, the other picture is from North America, and you look at this picture and you know that they have different ancestry, alternative ancestry, and you look at the picture and you can see they have different phenotypes from one another, and they of course have different genotypes as well. So that's colonies of ants. Now let's look at colonies of people, okay? These are my people. These are my great-grandparents who actually, this picture was taken in Fürth, which is about a 2-hour drive north of here, and my family was from Fürth. And so if you look at these, so these are Heinrich and Blanca, and these are their 3 sons, okay? And you can look at them, you can see in their faces, there's common ancestry here, right? I told you that, right? And so they have similar genotypes, and you look at their faces and they have similar phenotypes. And if you look deeply into their eyes, and you look deeply into my eyes, you can see similar neuroses, okay? And so because of common ancestry, we have common genotypes, common behavior, common traits, okay? And, but then if you want to see different genotypes and different phenotypes and different behaviors, well, again, we'll go to another continent. So this is an Inuit family. Both pictures were taken about 100 years ago. So this is exactly on the other side of the globe from the first picture. One was in Fürth in Germany, and the other was in Alaska. And this is an Inuit family. And of course, they have different ancestry from the first picture, and different genotypes, different phenotypes. And this kid probably has different neuroses than I have because, you know, it has different ancestry, right? Okay, so if we want to have alternative traits and alternative genotypes, we go somewhere else and we find different ancestry. But let's take this a step further, okay? So we said common ancestry gives rise to conserved genotypes, conserved phenotypes. When you think of a phenotype, you typically think of metabolism, okay? But on a broader scale, phenotypes are behaviors. Well, if you think about that and you take it a step further, it's not just all of the members of a family or all of the ants in a colony of ants, but Darwin told us in the 1800s, Darwin proposed that all life on Earth has common ancestry, right? I mean, the fundamental ideas of evolution is that life, well, you all know this, right? So there's common ancestry for all life on Earth. Well, when Darwin proposed this was a crazy idea, okay? But in modern times, of course, it's no longer a crazy idea. We have data, okay? And we sequence lots and lots of genomes, and sequencing of these genomes shows us that there's common genotypes, and there are similar phenotypes in many ways, metabolically, whether it's you or me or the ant or E. coli, we have certain metabolic processes in common. Those are similar traits. And we can see the connectivity, the common ancestry of all of life. And people have looked at this and said, okay, oops, sorry. you've seen trees like this where all life on Earth, and again, you, me, cow, ant, whatever, have arisen from what's often described as a last universal common ancestor, okay? So here we have a situation where it's not just all of the ants in a colony, but all life on Earth has common ancestry. And so if it all comes from the same origins, from this hypothetical Luca, and then all life on Earth is going to share similar components, similar traits, right? We all metabolize sugars in pretty much similar ways, okay? We all use the same 20 amino acids, all right? So if you hear that, and if I tell you there's this relatedness, there's this commonality for all of life on Earth, and if you're a really smart, creative young scientist who's trying to do new things, you go, huh? Are there no alternatives, right? I mean, you guys in your projects are always looking for alternative ways of doing things. And here I'm telling you that all life is related because all life has common ancestry and it's all doing it in similar ways. And so an intelligent, creative thinking person is going to say, huh? Aren't there alternatives? Aren't there other ways of doing this? Okay? And think about that, right? We often don't think about, we all think of making alternative in your projects in iGEM, but we're all still working with the same building blocks that came from common ancestry. So what if you want alternative ancestry, right? I said, this guy is saying, huh, no, I don't like that. Let's find an alternative. Well, if you want alternative ancestry because you're interested in alternative genotypes that are not related, and you're interested in alternative metabolisms or behaviors that are not related to last common ancestor, then what do? Well, you can't just go to another continent, right? I mean, we did that for my relatives and the Inuits, and we did that for the ants. And normally if I think I want something alternative ancestry, I go to another continent, I go somewhere else, different ancestry. But if Darwin tells us that all life on the planet arises from common ancestry, how do we find alternatives? So NASA, the space agency, has got this program on astrobiology. I think you've probably all heard that word. But the idea is, well, if I want to look for alternative ancestry, I have to go to alternative planets. Well, I'm not doing that. There's another approach, and that's what you guys think about, and that's to try and make alternative ancestry using some kind of synthetic biology. But in this picture, what the guy is doing is he's cutting and pasting DNA, right? It's a cute picture, right? There's even money on the table. It's a cute picture, right? So he's cutting and pasting DNA, he's moving genes around, but in a sense what he's doing is he's still working with the common ancestry because the DNA that he's working with, he mostly got the sequences. He may have ordered the DNA from a synthesis company, but the sequences arose from what Darwin described as the common ancestry of all life on Earth, okay? So in a sense, while this is synthetic biology, it's not alternative ancestry. It's somewhat beholden to the ancestry that we all share with all life on Earth. But these are the two options, right? And so most of us in this room are going to think about this option. And I just recently wrote a grant proposal to NASA about projects that I'll talk about later perhaps. And I thought, well, okay, when you write a grant proposal, you always want to say, this is what I'm going to do. But if it fails, I have plan B. So my grant proposal was about alternative lifestyles, alternative ancestry. So this is what we're going to do, synthetic biology. My alternative, if it fails, plan B is we got to go to another planet. Because there's really only two ways of doing this, right? If you want alternative, different ancestry from life on Earth, you have two choices. You go to non-Earth, or you make it from scratch, okay? And so if we want alternative ancestry, we either go someplace else, okay? Or we make it de novo. We make novel sequences and novel traits, okay? And so that's sort of how I'm going to try and introduce the concept of novel proteins. So when we think of novel genotypes, phenotypes, traits, and so forth, we have to think about the genes. You all think about genes. We have to think about the DNA. We have to think about the proteins. I'm going to just say I don't care about the genes, because if I can figure out proteins, anybody in this room can code for the protein, right? That's trivial, okay? So the deal now is, can we make novel proteins that never before existed, not just in my family in Furt, and not just in the ant colonies in Africa, but never existed on Earth, okay? So novel ancestry or non-ancestry. Can we make novel proteins? So we think about that and we say, all right, sequence space, what possible protein sequences can there be? So this morning we heard about using non-natural amino acids. I'm going to keep matters simpler. I'm going to say if we just stick with the 20 natural amino acids, okay, how many possibilities are there? Well, for a small protein, 100 amino acids long, there are 20 to the 100th possibilities. Okay, so let's imagine everybody in this room is working on this project, and we're making all of the possible sequences. And we're iGEM people, so we work really hard. We're in there 24/7. We're making all those proteins, right? We're going to make all of them. And we're going to put all of these sequences into that flask. And if we did that, all of us, right, and we're really good, and we made one molecule of each of those proteins, the size of this flask would have to be not just larger than the known universe. That's nothing. It would have to be larger than a mole of universes, okay? Just one molecule of each. Okay, so sequence space, the number of possible sequences is staggering. We can't make all of them, okay? So that's the hypothetical sequence space. But then along comes Darwin, or along comes nature, and evolution occurs over the course of, on this planet, let's say, well, 4.5 billion years. So we've got billions of years, and this is the hypothetical starting material. It wasn't all there, of course. There are not enough atoms in the universe. But from this hypothetical starting material, with powerful selections over billions of years, we end up with something spectacular, life, okay? And life is sustained by not that many sequences, right? E. coli has 4,000 sequences. You have about 250,000 genes, 25,000 genes encoding, depending on splicing, quarter of a million proteins. I look at that and I say, well, these are tiny numbers compared to the possibilities. So I look at that and I say, oh, these sequences must be really special because they were weeded out of an enormous number of possibilities, and yet they do something really special. They enable life. So I look at that and I say, can we do that? Can we actually make entirely novel sequences completely from scratch, no common ancestry with life on Earth? Can we do that and make artificial proteins that make artificial proteomes and are encoded by artificial genomes? And if we can do that, then we're thinking about alternative lifestyles. And I kind of like this idea of alternative lifestyles. Normally think of, hipsters living in the city or whatever they're doing, but I'm going to think about alternative lifestyles in a much broader sense. I'm going to say lifestyles from a metabolic point of view of all life on Earth are related, right? More related than your lifestyle with your parents' lifestyles, right? All life on Earth has related lifestyles. Can we now make novel sequences and bring up the concept of alternative lifestyles? So how do you do that, right? You say, all right, we're going to make lots of them, all right? So the number of possibilities is beyond comprehension, okay? However, if I make them randomly, in a totally combinatorial random way, most of them are garbage. Random sequences don't fold. So I'm tempted to explore sequence space by making random combinatorial libraries, but I don't want to do that because the grad students in my lab are going to have a fit because most of those sequences won't fold, okay? So I want to impose some sort of rational design. I want it combinatorial, right? But I also want some sort of rational design so most things fold, okay? So here's protein folding for babies, okay? This is the easiest protein folding slide you will ever see. Yellow is olive oil, it's hydrophobic. Red is cranberry juice, it's polar, it's hydrophilic. Does not dissolve in water, dissolves in water. If you have liquids, you can get phase separation. We all know that, right? But if you connect the beads on a string, there's no phase separation, they can't separate, and so they have to fold. You all know this, I'm not telling you anything you don't know, but it's a nice picture, right? And so the hydrophobic effect is the driving force for protein folding, and this is well known for a long time. So if this is what calls the shots, we said, let's see if we can make a combinatorial library of many, many, many novel proteins, novel sequences, but all of them are biased or pre-organized or pre-chosen in such a way that they'll fold. So if you think of this picture where yellow is, for everything I show you now, yellow is going to be olive oil, hydrophobic, red is going to be polar, water soluble. Okay, and we say, all right, suppose we want to make proteins that look like this. Okay, this is a bundle of 4 helices, and we said if the hydrophobic effect is what's the most important thing going on, then all I have to do is put wherever I want an amino acid facing the center. This is echoing, is this okay? You okay? Can you hear me okay? Somebody tell me from the audience, is that going, it's okay? It's okay, all right, the back of the room said it's okay, so, sorry?
Just put it on.
Okay, so we said that all we have to do, if it doesn't bother you, it doesn't bother me, it's fine. So all we have to do is wherever I want an amino acid pointing toward the center, it has to be yellow, hydrophobic. Wherever I want it pointing toward the outside, towards water, it has to be polar, okay? And so we decide, okay, this is a binary patterning, okay? It's just yellow and red, it's binary, it's 2 things, right? But then we looked at the genetic code, which you've all looked at many times, and we looked at the genetic code and we realized, aha, this binary patterning, this binning of yellow and red, olive oil and cranberry juice, fruit juice, whatever, already exists in the genetic code. Many of you may have noticed that before, but the codons that go NTN, okay, are all hydrophobic, and the codons that go NAN, forget that box, are all polar, okay? And we said, aha, so nature knows about this, okay? And we said, if we want to make a vast combinatorial library of proteins that look like this, none of them are related to anything in nature, these are sequences we're making up from scratch, then all we have to do is pattern the polar and non-polar amino acids in this binary way. So we said, okay, let's just write this down, okay? Wherever there's a yellow circle, that's olive oil, it's greasy. Wherever there's a red circle, that's fruit juice, it's polar, okay? And this is done in the patterning, the same patterning as this, okay? It's the same patterning here. We have certain amino acids are fixed, right? Methionine, tyrosine, glycine, but most of the alpha helices are combinatorially diverse, where that yellow position can be leucine, isoleucine, valine, anything hydrophobic, and the red position can be any polar amino acid, arginine, lysine, whatever you want, okay? And we said, and then the blues are these turn residues. And the hypothesis was that this binary patterning that's connected to the hydrophobic effect is so dominant that I can make all the mistakes I want and they'll still all fold into structures that look like this. That's a crazy idea, right? So we're not doing all of the chemistry that you learn about. We're not designing van der Waals interactions or hydrogen bonds. We're just doing it like you would do with crayons. You know, it's red here and it's yellow there and off we go, right? They're all going to fold. Okay? So we did this. So we make a large library. So the non-polar codons are what I said before. Polar codons, turn codons, and so on. These are combinatorially diverse. You notice the ends and the, semi-randomization. And this is not a computer experiment. This is a wet lab experiment, okay? So now you have 10 to the 6 different sequences in the freezer, and they're all done by making synthetic DNA. You all know how to do this, except using combinatorially diverse codons, where you have an N at certain positions. So you have a million different sequences, all having that same pattern, but each one a different sequence. And then we say, aha, this is a model proteome. This is a proteome, a collection of proteins that shares no ancestry with anything in nature. So it's a possibility of alternative genotypes, alternative phenotypes, okay? It's de novo. And so that's our artificial proteome. We make it at the DNA level, we put it in E. coli, we express it. I think you all know how to do that, okay? And it's now an artificial genome, because these are artificial DNA sequences, we'll call it that, an artificial genome, coding for an artificial proteome, novel protein sequences. And then we say, all right, can I actually do this to sustain life, right? Because that was the goal at the outset. We're thinking about alternative ancestry, alternative lifestyles, if you want. And so is this library that I just described to you up for the task? Well, there's 3 things you want to ask. So now we're going to get into more of the experimental part of things. So is this library sufficient to arrogantly call it an artificial proteome? So 3 things I want to ask. Do these sequences fold? Remember I told you a few minutes ago that if you make random sequences, most of them don't fold. So the first thing I have to convince you is that this binary patterning of yellow and red is sufficient to promote folding. Is that true? Do they have any kind of activity? Do they bind? Do they catalyze? Do they do anything? Do they look like proteins? Anything? And most importantly, what I want to talk mostly about is, do they function in vivo? Can we actually get these things to support life? Okay, fold into stable structures. Here's some structures we've solved. These are NMR structures. This is number 824, number 836. I'll turn the lights down just for a second because these are pretty pictures. Yeah. Okay, so you can see here, this is the 4-helix bundle. If I turn it on end, the yellow greasy stuff is all on the inside. The polar stuff is all on the outside. Sometimes we get dimers, but we basically get what we're expecting to get. Okay, we get crystal structures or NMR structures that are in fact folded. So yes, by the simplistic idea of patterning yellow and red, we can indeed generate sequences that fold into structures. And remember, they have no similarity in sequence space to anything in nature, okay? So number 1 was structure stability. Yeah, we can make some really stably folded proteins here. Number 2, biochemical activity in vitro. Ask me some other time. It's a short talk. I don't have time to go into it, okay? But yeah, we can do that. But the really important thing is, can we function in vivo? Can novel proteins sustain the growth of a living organism, okay? And so that's a profound question, right? It's almost theological, right? Can you make sequences, can humans make sequences in a laboratory that sustain life? And I should say, I spent a lot of time talking to theologians and philosophers, right? Not just chemists and biochemists. And this is, this is sort of at the interface of the idea of humans creating things that sustain life, where those things have no ancestry to what came before. So it's a profound question, but this experiment is really, really easy, okay? Here's the experiment, okay? You take a cell, this is all in E. coli, you delete an essential gene. We all know, you all know about that, right? You make a strain of E. coli that's delta XYZ. And so here we go. Here's our collection of 1 million of these sequences. Again, it's combinatorially diverse. I have any hydrophobic at the yellow, any polar amino acid at the reds, combinatorially diverse. I have 1 million of them in the freezer. Here are 6 of those million. They fold into these 4 helix bundles. 5 of them are gray, and 1 of them is, purple, okay? Then you take cells, so you have billions of E. coli cells. You have a test tube full of E. coli cells, right? And they're all missing one essential gene, okay? They're missing a gene that they need in order to grow on, let's say, minimal medium, okay? If you put these cells on a plate of minimal medium, nothing grows, okay? Because they're deleted for an essential gene. Now what I do is so here are the cells, and then they're deleted, okay? I put in a plasmid that perhaps, hopefully, codes for a protein that can rescue that deletion. The white ones don't, the purple one does, maybe. And then you put that on a petri dish of minimal medium, and if you get a colony, whoa, that says, providing all the controls work, which we all know about that, everything looks great on slides, but in the lab, it doesn't always work. But suppose it does work, right? Suppose it does work, okay, then what you see here is a colony of cells that would not have grown except for the fact that they received a sequence of DNA unrelated to anything in nature that can support the growth of a living cell. And the cool thing about this is you can do this, so E. coli has 4,000 genes. You do this for any deletion in E. coli you want. And so here's something that iGEM people are going to love. There are 4,000 genes. You can fail 3,999 times. But if it works once, it's really cool, okay? So you do this over and over again. We've done it many times. Most of the time it fails. Word to the wise, most experiments fail, okay? But sometimes it works. So here's one that worked. So minimal media plates, delta ILV, the gene is involved in isoleucine biosynthesis. We put in control DNA on a plasmid, nothing grows. We put in a library of 1 million novel sequences, you get colonies, okay? The picture's not all that pretty, but it says there's a novel sequence that comes from non-natural ancestry, that has no ancestry in a sense, that is sufficient to sustain life, okay? Let's do it with another knockout. This is delta Fez, I'll tell you about what that is later. The controls failed to grow. The library gave you many different sequences that were able to sustain life, okay? Again, despite the fact that they're not derived from natural living systems, okay? So here are some of the things we've been able to, this is more detail than I want to go into in a short seminar, but there are various different knockouts. You can sort of read this quickly while I'm talking. Various different knockouts where we've deleted this, or this in E. coli, and we've come up with de novo sequences, entirely artificial sequences that can rescue the deletion. In other words, artificial sequences unrelated to anything from Darwin that can sustain the growth of a cell, okay? And then you look at these, okay, so these are these 4 that we've got, Fez, CRB, blah, blah, blah. And so here are the crystal structures of the E. coli proteins themselves. They're big, they're complicated, and here's our dinky little 4 helix bundle, okay? So proteins from a library of these can, in some cases, often it fails, but when it works, can rescue the deletion of these complicated things, right? So how do they work? How do the SYN proteins, the SYN proteins are these synthetic artificial proteins, how do these SYN proteins rescue the deletion of an enzyme, okay? So it's kind of bizarre that it worked at all, and we were pretty amazed that it worked at all. But then you have to ask the harder question of how does it work? And you can imagine there's 2 possibilities. Either my SYN protein, my artificial protein, catalyzes the same reaction. Okay, that's cool. That's sort of what you expect naively, right? It's a simple idea. It's hard, but it's what you expect. But alternatively, the bottom box says maybe the synthetic protein somehow tweaks the natural system in one of these ways, that way. So which is the answer? Yes, okay? I'll show you. So first, can we catalyze the same reaction? So let's look at Fez. So delta Fez, so here's what Fez does, okay? Fez is, Fez is the name of the gene, encodes enterobactin esterase. Let's explain what that is. This is enterobactin. You can ignore the colors now. The colors just say it's threefold symmetric. It's a small molecule that binds iron very tightly. E. coli sends that small molecule out into the medium. It binds iron with a crazy tight binding constant. E. coli then imports that back into the cell, and then it has the iron in the cell, but it's bound to this siderophore, this enterobactin. And then E. coli has an enzyme, the esterase, that breaks it into pieces and liberates the iron. Great. If the E. coli is delta Fez, then it's dead on minimal medium, on low iron medium. Then it's dead because it gets the iron into the cell by this system, but it can't get the iron out of this molecule here, because the white arrows don't exist. Okay? So this is an essential gene for E. coli growing in low iron conditions, and the E. coli are dead. You can put zillions of cells down on the plate, they're dead. But we come in with an artificial protein and it rescues. So we have an artificial protein that is rescuing the deletion of Fez. Okay? How does it work? Well, we did a bunch of things. One thing is if you grow it on high iron conditions, okay, if this accumulates, this iron-bound stuff here, the colonies are red. If you have the wild-type esterase or our novel protein, synthetic esterase, this stuff is broken into bits and the red color goes away and the cells are more healthy and bigger. In vivo. In vitro, you do the HPLC, you do the mass spec. I won't bore you with all the details, but you can break down this peak into the other ones. This is the HPLC. Point is, what we now have is basically this protein was really called 4F2. It's an esterase, okay? So as far as I know, this is the first example ever of a completely novel sequence that behaves as a life-sustaining enzyme, okay? And then what we did is we did sort of a cool experiment that I wouldn't have thought of, but the best experiments in my, all right, here's the story. Best experiments in my lab are always like this. Student comes into my office and says, I'm gonna do da-da-da-da-da-da-da. Michael says, that'll never work. A year later, we have a paper, it's great, okay, right? And so she said, well, I know somebody who made the non-natural enantiomer of this, and I'm gonna challenge it with that, and it didn't hit the enantiomer, right? So our novel enzyme is life-sustaining and stereospecific. It only works on the natural enantiomer. And we then moved it from the plasmid onto the chromosome, and so it works when it's expressed. So we're thinking about towards artificial genomes, right? We're making novel sequences on the chromosome, replacing natural sequences with a novel sequence that has nothing to do with anything that evolved from common ancestry, and it's life-sustaining. This just came out a few months ago. The title is really all you need to see. So it's a novel enzyme. Yeah, you can read the title. Okay, so in that case, we have a novel sequence unrelated to common ancestry that is life-sustaining as an enzyme. But going back to here, I said, well, there's really two possibilities. Either the proteins could function as enzymes, as catalysts, or they could do something in terms of playing around with the natural system, changing expression or activity. So let's show you one of those. So here's central metabolism, and you remember your biochemistry, or if you happen to be working on this, remember that there's a simplified version of the Tca cycle. Do you remember this? It took me a while to remember it. I haven't thought about it in a long time. But remember, you get into the Tca cycle with acetyl-CoA and oxaloacetate, make citrate, and then you do all the stuff, and then something has come out off the side to make other stuff. Okay, that was a really very technical definition of the stuff. Okay, so you cannot do a genetic selection for the Tca cycle because E. coli can get its energy and carb metabolism in other ways. But you can do a genetic selection by selecting for glutamate, because glutamate comes from here, and no Tca cycle, no glutamate. So we select on minimal plates to rescue a delta GLIT-A deletion because it can't make glutamate. But it's really asking, is the Tca cycle crippled or not? Everybody got that? Okay, so that's the logic. And we get a protein called syn-GLIT-A. It's a synthetic protein, one of our 4-helix bundles, that rescues GLIT-A. Okay? And then we play around with it. We say, but it does not rescue this step over here in a different strain. So it's not providing a bypass pathway to glutamate. It's coming from here. Okay, so we have a novel protein, and lots of other control experiments. We have a novel protein that enables this step to happen in a cell where that was crippled and it couldn't happen. Okay? A novel protein unrelated to anything in nature. All right? And so basically this sort of summarizes what I was trying to say, all right? So you figure, okay, great, it's an enzyme, right? Just like the one I told you a few minutes ago. So you purify the protein, and oh my goodness, it doesn't work, okay? And so your immediate response is do it over again, and do it over again, do it over again, do it over again. Then your final response is this, okay? like, this doesn't make any sense, okay? How does this novel protein rescue the deletion of the enzyme citrate synthase if it's not an enzyme itself? So Katie, who was a very bold and terrific student, said, well, let's do RNA-seq. For those of you who don't know RNA-seq, we're asking, how is RNA expression changed in a cell when I add this synthetic protein? And I won't go through all the details, but various things are turned up manyfold. And some of them are colored in pink, and I want to draw your attention to one of the arrow, PRPC catalyzed this reaction. Look at this. It's not citrate synthase. It's a different pathway. It's a different chemistry. It's 2-methyl citrate synthase, which normally doesn't function in the Tca cycle, okay? But it turns out that's a promiscuous enzyme. You all know the word promiscuous? No, not that one, okay? But you know the promiscuous here, right? And so it's a promiscuous enzyme, which means that if you make enough PRPC, and it has a tiny bit of promiscuous activity, it will do the rescue. Like I say, we're making a ton of it, 32-fold more than natural, and so that rescues. And so in this case, we have a novel protein unrelated to anything in nature that enables central metabolism to happen through the Tca cycle, right? This is central metabolism, it's kind of important, okay? By upregulating something else, alternative lifestyles, alternative metabolic pathways. So what I'm showing you here is we've gotten rescues, we've got novel proteins unrelated to something in nature, where in this case, it's rescuing by some rewiring of gene regulation. And in the Fez case, it was rescuing by actually acting as a bona fide enzyme, okay? Let me close up with the last 2 or 3 slides, putting this all together and saying, okay, we want to look at what I'm calling alternative behaviors or alternative metabolisms, alternative genotypes, alternative ancestry, okay? And so from that, I want to think about making new genomes and new proteomes. And again, yeah, new genomes and new proteomes. So thinking of an artificial proteome, so here's what we did. We said, all right, we have, E. coli has 4,000 genes. Lots of them are dispensable. If you're not trying to grow on lactose, you can delete the lac operon and who cares, okay? But there are 400 that are absolutely essential to grow on minimal medium, okay? We have 4, let me not go through that yet. Remember, we had 4 rescues that we showed you before in 4 different pathways. GLID-A, SIRB, ILV, and Fez, okay? So what we now do is let's make an E. coli strand delta quad. It's delta deleted for all 4 of those at the same time, okay? It's a very sick cell. It cannot grow on minimal medium. In fact, it's dead to the 4th power, right? Any one of these deletions would prevent growth on minimal medium. But this one's deleted to the it's dead to the 4th power. Then you come in with a control plasmid, nothing is going to grow. If you come in with DNA encoding these 4 different artificial proteins that I isolated in earlier experiments, the question is, will that grow, okay? And so here's the deal. The strain is delta quad, no colony on minimal media, it's dead to the 4th power. And here it is, it comes in with 4 novel proteins on a plasmid, and it's working, you get colonies. They take a week to grow, it's not a happy cell, okay? But it grows, okay? So what we've said now is we have 4 novel genes encoding 4 novel proteins. Well, 4 out of 4,000, it's 0.1%, but there are only 400 that are essential. So 4 out of 400 suggests we've actually replaced 1% of the essential genome, okay? And so now we really have to start thinking about the possibility of not just synthetic biology at the level of moving genes around from one organism to another and rearranging them, but synthetic biology from the perspective of can we use genes and proteins that did not evolve from common ancestry, but nonetheless have the ability to sustain life. And I'm going to summarize that here, right? So we can make artificial proteins that can sustain life either as enzymes or as doing gene regulation. I told you about that one, I told you about that one. Got another one I didn't tell you about. Synthetic biology, right? That's what you guys all do, right? So synthetic biology, you guys in iGEM often talk about parts kits, right? They're parts, they're modules, they're parts kits, okay? And you order them up or you make them. But here's the thing, the parts kit don't have to be limited to those parts that evolved in nature, okay? And that's special for you guys, right? Because most of you are taking genes and proteins and regulatory elements that you're getting from nature and rearranging them, putting them in different places. But in fact, these experiments suggest that your parts kit need not be limited to those that arose from Luca. You can make parts kits from scratch. Point 3, this was a baby step, right? All we did is we looked at a bunch of deletion mutants. We didn't look at all 4,000 in E. coli. We did not have 3,999 failures before we got a success, although in principle you could. We only looked at a bunch of them, and we've only made 1 million alpha helical proteins. I mean, that's just a really small number, right? You should be able to make gazillions of them, alpha proteins, beta proteins, all kinds of stuff. So it's just a baby step. And finally, the concept of life reinvented. Eschen Moser, a very famous organic chemist in Tca, made this comment once saying that the origin of life cannot be discovered. It has to be reinvented. And so I've sort of played around with that quote a lot. And so the idea of life reinvented, it suggests that artificial genomes that sustain life, I mean, we replaced 4 in E. coli. That's just a weird E. coli with 4 weird genes replaced with artificial sequences. Okay, it's just a weird E. coli. But if you did 400 or if you did all 4,000, then it wouldn't just be a weird E. coli. You would have to think of it as a new form of life. And so the question is that possible? And that's really not the question. The question is when that's possible, right? That is something that's going to happen. Okay. Okay, I'm going to close up with thanking people. So these are the people who worked on the project. I've coded them in a way that you see, that's a postdoc, that's a graduate student, and lots of undergraduates. So be inspired. There are lots of undergraduates who did this work and who co-authored much of this stuff. Terrific undergrads, and yeah. And this undergrad is particularly terrific. He was an undergrad in my lab many years ago at Princeton, and he's now a colleague on the faculty at Princeton, so that's just great. So that's who did all the work, and this is who funded it, and I thank you for your attention. So.